In this video, you will learn about the structure and function of neurons. These fascinating cells process information in the nervous system. You will also learn about the structural and functional classification of neurons. First, we will focus on the most common structural class of neurons that have a multipolar shape and a wide range of functions. These cells are found in the central nervous system, where they form the neural circuits that produce conscious thought and allow us to regulate the movement of our bodies. Finally, we will study examples of sensory neurons in the body that have bipolar or pseudo-unipolar structures. Let's begin by describing the structure of a typical multipolar neuron, which is the most common type found in the central nervous system. The soma, also known as the cell body, is the central part of the neuron. It contains the nucleus and organelles that maintain the cell's metabolic activity. Extending from the soma are dendrites, branching structures that act like antennae. These dendrites are covered in receptors that detect neurotransmitters released by other neurons. At the base of the soma is the axon hillock, a tapered region where the cell body transitions into the axon. This is the site where an electrical impulse called the action potential is initiated. The axon is a single, long process that carries the action potential away from the soma toward other neurons or target tissues. Surrounding the axon is the myelin sheath, a fatty layer formed by glial cells. This sheath acts as insulation and greatly increases the speed of action potential conduction. Along the axon, there are nodes of Ranvier, which are small gaps in the myelin sheath. These gaps allow the action potential to jump from node to node in a process called saltatory conduction. The internodes are the myelinated segments between these gaps. At the far end of the axon are the axon terminals. These are the structures that release chemical messages called neurotransmitters. The synapse is the junction between the axon terminal and another cell. In this illustration, the synapse forms between the axon terminals of one neuron and the dendrites of another neuron. Now let's move on to the classification of neurons, both by their function and structure. Neurons can be grouped based on the direction in which they carry information. Sensory neurons, also called afferent neurons, carry information from sensory receptors toward the central nervous system. These neurons allow us to detect stimuli from the external and internal environment. Interneurons, also known as association neurons, are found only within the central nervous system. They serve as connectors between sensory and motor neurons and are essential for processing and integration. Motor neurons, or efferent neurons, carry commands from the central nervous system to effector organs such as muscles and glands, allowing us to respond to stimuli. Neurons also differ in their physical structure. Multipolar neurons have multiple processes extending from the cell body. Here we see two different examples of multipolar neurons found in the brain. While these are both functionally classified as interneurons, most motor neurons also have a multipolar structure. Most of the neurons in the central nervous system are multipolar. Multipolar neurons have numerous extensions from the cell body. Most of them are dendrites, but one is the axon. Pseudo-unipolar neurons have just one extension from the cell body. However, this extension quickly branches into two parts, one that functions like a dendrite and another that acts as an axon. Bipolar neurons have two distinct extensions attached to the cell body. One extension is the dendrite and the other is the axon. Bipolar neurons are found in specialized sensory organs. Smell, vision, and hearing are examples of special senses that involve bipolar afferent neurons. For example, in the olfactory epithelium, bipolar olfactory receptor neurons detect odorants in the superior nasal cavity. Bipolar cells of the retina in the eye are another example of an afferent neuron in a special sensory organ. Bipolar cells in the retina receive visual signals from photoreceptors. Bipolar afferent neurons are also found within the cochlea, a structure of the inner ear. In the cochlea, bipolar cells of the spiral ganglion receive auditory information from hair cells. Pseudo-unipolar neurons are typically found in the peripheral nervous system and are responsible for transmitting sensory information. Most sensory neurons have this structure. Good examples include the sensory neurons for touch that have their dendrites in the skin. Let's review with this example of a person adjusting the temperature of water in the shower. The sensory receptors in the skin are the dendrites of the afferent neurons. This afferent neuron has a pseudo-unipolar shape with the cell body found in the dorsal root ganglia. The axon of the afferent neuron enters the spinal cord and extends up to form a synapse in the brain. A circuit of interneurons in the brain process the sensory information and send commands out to the motor neurons. So far, the motor neuron and interneurons in this example all have a multipolar structure. The thermoreceptor afferent neurons carrying information about the temperature of the water have a pseudo-unipolar structure. If the person is using vision to help guide their movement, the bipolar afferent neurons of the retina will relay information from photoreceptors in the retina to a circuit of interneurons with a multipolar shape. You can see from this example that everyday activities require us to use many different neurons communicating through synapses. The dendrites of afferent neurons detect sensory information. Information travels through the axons of neurons in the form of action potentials. When the action potential arrives at the axon terminals, neurotransmitters are released to communicate with the dendrites of interneurons in the brain. When an action potential travels down the axon of an efferent neuron, the axon terminals will release neurotransmitters at neuromuscular junctions to stimulate contraction.